to our last interactive session. We're going to have dinner, of course, after this. And this is going to be the Candida group. I'm going to co-host this with, or co-chair this with Louise Ostrowski-Zeitner. Now, we've had a little bit of a change in the agenda. Two things that I'll mention. First, Michail Lionakis, who's going to be our first speaker now. His wife is probably going to deliver tomorrow. So he thought that in order to keep his marriage together, he might better stay close to home. So she is, I think she's bedbound right now, but he's speaking to us from his home or his office in Bethesda. And then at the end of this session, tacked on, Tom Chiller is going to give us a 10 or 15 minute update on Candida auris, a talk that Luis and I heard when we visited recently at CDC. So that'll be tacked on to the end of this. I've asked our speakers to try and keep their comments limited to 25 minutes so that we don't run over, and have plenty of time for open discussion at the end of the session. So let me, so Michail is of course going to join us here electronically. So Michail is a physician scientist who obtained his MD and degree from the University of Crete in Greece. He then did his clinical training in medicine and infectious diseases and research training in mycology and immunology at MD Anderson Cancer Center. In 2012, Michail was recruited as a tenure track investigator at NIH, where his bench-to-bedside laboratory research focuses on understanding the cellular and molecular basis of host defense against mucosal and systemic fungal infections in mice and in humans. So, Michail, welcome. I'm sorry you can't be here. You're missing some great weather, but glad that you're going to be a father tomorrow.
Thank you very much, Peter, and everybody. I hope you can hear me well.
We can hear you just fine. That's perfect.
Perfect. So, what I wanted to do today is, as Peter mentioned, in my lab, we try to understand how our immune system responds to mucosal or systemic challenge by fungi. We focus on Candida, which is the most common human fungal pathogen. And our goal really eventually is to devise mechanism-based strategies to develop better diagnostics, therapeutics, and risk stratification strategies for patients with fungal disease. So today I wanted to use some of the data that we've generated, and also some of the data that others have generated, to introduce, I think, a timely question for us as mycologists, which is, I think the time has come to develop trials in mycology, which would be pioneers in precision medicine. And I'll base my talk around that today, and you'll see how that goes. So I don't think I need too much to discuss with you about what's the burden of systemic candidiasis, Candidemia, and deep-seated candidiasis in humans. It's the leading cause of nosocomial bloodstream infection in the US and in the developed world, with a lot of cases, dozens of thousands of cases per year, with a lot of cost. And mortality that has remained 40%, despite the fact that over the last 10, 20 years we have better drugs. Our excuse of amphotericin unbeing a very user-friendly drug has really not been there anymore. So what that really means is that this is obviously an unmet medical condition, and better understanding of its immune responses could lead to two potential outcomes and better outcomes. One is, by better understanding immunity, we can develop immune-based therapies to augment or supplement antifungal therapy. But importantly, what I wanted to talk today about is, you all know that if you take 100 patients in the ICU that would be at risk for developing Candidemia and deep-seated candidiasis that have similar clinical and microbiological factors to put them at risk for that, only about 5 or 10 or less will develop the disease, which would suggest that there are some patients that are uniquely more susceptible from an immunological and genetic standpoint. And if we can understand better the immunity, we could potentially identify those patients in whom either prophylactic antifungals or Jack Edwards vaccine, which I think Jack is talking after me, might be applicable to then develop personalized means of protecting them against a very, very lethal infection. So how do we go about that? We use the commonly used mouse model of the infection that mimics what happens really in the majority of the cases, as far as at least the portal of entry, which is the skin. In this model, kidney is the target organ of the infection, and if we look at autopsy studies of non-neutropenic patients with disseminated candidiasis, that also appears to be the case. And what we know about this disease from a host standpoint, both in mice but also in humans, is that neutrophils and monocyte macrophages are critical, but not lymphocytes. So patients with AIDS or SCID patients don't develop this disease. And what needs to happen for an effective immune response is these cells need to traffic to the site of where Candida is and become activated. And a few years back when I was at Phil Murphy's lab as a postdoc, I became very interested in these molecules called chemokines, because what they do is they do trafficking and activation of cells by acting through their chemokine receptors. And an important concept in immunity is that any immune response is really the balance between, on one hand, you want to kill the invading pathogen, in this case Candida, but at the same time you want to minimize the tissue damage that is caused by the immune response itself, so that when you deal with an infection in the kidney, you're left with the kidney after the infection is controlled. And why is that important? Because if we identify factors that mediate immunopathology, we could potentially look at new targeted therapeutic interventions for the disease. And if we identify factors that mediate protection against the infection, as I'll show you in two examples today, if we identify correlating SNPs in humans that have similar loss of function alleles, we might actually look at factors that might look at those patients who are at higher risk to develop the infection among the pool of all of the patients. So what we've done in my lab over the last few years, in a summary, through a very unbiased screen of the entire chemoattractant system that has more than 70 tools, is so far we've shown that CX3CR1 protects and mediates resistance by promoting macrophage survival, that CCR1 is a mediator of immunopathology by promoting excessive recruitment of neutrophils to the infected tissue, and that CXCR1 mediates resistance by promoting neutrophil killing activity. Now, in 2 out of these 3 cases, as you can see, we've uncovered unconventional roles for chemokines, which were initially thought to mediate trafficking of cells. So I would urge everybody who studies immunity to think of these molecules not just as trafficking mediators, but as mediating multiple immune functions in general. Now, in 2 out of these 3 models, we've been able to translate this directly to humans, first at the population genetics level, taking great advantage of 2 great collaborators, so John Perfect, who is there with you, and Michail Netea, who is in Nijmegen, where at the population genetics level, we've found alleles that are loss of function that confer susceptibility in ICU patients. And then in our lab, we've also been able, in primary human cells, to confirm the mechanistic biological defects in these primary human phagocytes. So today what I want to do is briefly mention these 2 as, again, a platform to introduce the concept that I think the time has come through not only these alleles, but other alleles that I'll also mention at the end of the talk, to potentially intervene in an individualized manner in the future. So I mentioned chemokines. Not all of you are familiar with them the way I am. So these are chemokines are a large family of cytokines that bind to G protein coupler receptors. And what they do is, through a concentration gradient, they mediate trafficking of cells from one place to another. And the place where they end up is inflammatory and infected tissue. And what they do is, through a complicated sequence of events, they make cells activated, they adhere, and they transmigrate through a gradient of an increasing chemokine gradient. Now, this system is quite complicated in humans. This is just putting it into a cartoon. There are now 74 chemokines and chemokine receptors, and we haven't discovered all of them. You're all aware of this system in HIV with CCR5 and CXCR5. We know about Daffy and the protection from malaria. There's a primary immunodeficiency called WHIM caused by a receptor deficiency in that. And today I'm going to talk to you about 2 alleles, 2 receptors where Candida belongs, CXCR1 and CXCR1. So starting with monocytes and macrophages, which are important host cells for this infection, almost 20 plus years ago, there was a paper that clearly showed that if you deplete monocytes, macrophages, and dendritic cells with clodronate, the mice don't do very well, so they all die, as opposed to mice that have intact mononuclear phagocytes. And surprisingly, there hasn't really been much of a follow-up study to that, so we didn't really know, is this monocytes, is it macrophages, is it resident or inflammatory cells, and what molecular factors really mediate these protective effects. So we took advantage of the fact that there are two prototypic chemokine receptors called CCR2 and CX3CR1. CCR2 is a marker in inflammatory monocytes and CX3CR1 on resident macrophages. And as you can see here, we found that CX3CR1 was absolutely critical for control of this infection. CCR2 had effects as well, but they were more modest, and I'll come back to that at the very end. And this was quite robust using multiple different mouse lines. And the reason why these mice were not doing very well is because they had a significant amount of fungal load in their tissues. Here's on the right side, these immunofluorescence pictures show D-tomato, Candida, and you see these big fungal balls in the kidneys of mice that lack CX3CR1. Now, to cut on directly to the chase, what we decided to do is use live imaging in mice that express CX3CR1 in mononuclear phagocytes, but taking advantage of the fact that the first few hours in the infected tissue, more than 90% of green cells are tissue-resident macrophages. So here you see 2 hours after infection, a green macrophage that has engulfed 2 red Candida yeasts in it. In the next picture, you'll see a almost 30 micron long filament of Candida with the yeast form of it that has been wrapped around by multiple macrophages. Now, this is quite striking because this is 2 hours after somebody put the Candida through a tail vein of a mouse. Hyphae has been, the yeast found its way to the interstitium of the kidney, and 30 micron long filaments have been already generated. For all of us that work with Candida, and we take Candida in a test tube and we put it in 37 degrees in serum, it takes about an hour and a half to make a 5 micron long germ tube. Again, speaking about why it's important to really look in vivo for immunity, this is really putting this in a different perspective of what really Hyphae priming conditions are. Now, the only time that we saw green and red interact with each other was the first 6 hours after infection, which was a great surprise to us. But the reason for that was because very quickly after the first 24 hours, Candida got into the tubules. And if I show you here a picture of the tubules, the surrounding being green, all of these macrophages are surrounding the tubules but are never inside the tubules. So all this black in the middle is where Candida is. It's the tubular space. So Candida has an immune evading mechanism, at least towards monocyte macrophages, to not be in contact with them beyond the first 6 hours after infection. Now, in this model, the reason why these mice died, because that interaction between Candida and macrophages was dramatically impaired. So again, looking in vivo in mice that lack this receptor, as opposed to about 90% of Candida structures that would be engulfed, as you see here on the left picture, by a macrophage, almost half of the Candida structures in mice that didn't have CX3CR1 were hanging out in the middle of the infected tissue without any green around them. And this was not because the macrophages had a problem with recognizing or binding to Candida. Instead, what the problem was that they were significantly decreased in numbers, both at steady state, but also throughout the course of the infection. Through a series of experiments, we showed that this was not because of impaired trafficking or differentiation or proliferation of the cells, but instead these cells actually were dying in the tissue. This is percent of dead cells as a function of whether they have the receptor or not. We were then able to show that there was an apoptosis defect in mice lacking the receptor. Now, chemokine receptors have emerged now as major modulators of survival in different cell types, and AKT seems to mediate most of these effects downstream of G protein couple receptor coupling. So we decided to look at that in the tissue, and indeed, when you out of the infected tissue, they have much more cleaved caspase 3, which is obviously a marker of apoptosis, and they're unable to induce phosphorylate AKT, which is again a critical pathway for anti-apoptotic effects through G-protein capture receptors. And if you look in the papers in Candida, we've kind of given macrophages a bad name as a community the last decades because we've used the wrong cell type. So if we take bone marrow-derived macrophages or peritoneal macrophages, it's actually pathetic in killing Candida. But if you do the cumbersome otherwise experiment to take macrophages out of the kidney, as you can see on the left panel, almost half of the Candida will be killed. And in the right panel, what you can see is that when we look as early as 12 hours, and we've looked as early as 6 hours as well after infection, at a time when no other cell has been recruited in the model in substantial numbers, there is already an increased fungal burden, suggesting again that macrophages are critical for protection. Now, we were able to take advantage of an allele that was published here at NIH almost 15 years ago in this receptor that was shown to protect against cardiovascular disease in the Framingham study, and work with John Perfect and Melissa Johnson at Duke and Mihai Netea in Nijmegen in 2 independent cohorts to show that this carrying this allele was an independent risk factor for developing the infection, this Candidemia in the ICU. And then worst outcome defined as having disseminated disease with an odds ratio of close to 3. The next question, obviously, that arose in follow-up unpublished data that I'm going to show you now is how does this work in humans? So in mice, we have a mechanism, and at the population genetic level, we have a mechanism, a correlating observation, but how do primary human cells behave? And a very talented postdoc that now is in med school in my lab was sorting monocytes from almost 120 healthy donors from RNA's blood bank that we genotyped in order to identify wild type and knockout patients. As you can see here by calcium flux, on the left side is the schematic and the right side is the compiled data. These monocytes indeed have about 50 to 60% impaired signaling through this receptor. So this is a true loss of function allele. That's not because the receptor is not expressed on the surface, but through a complicated series of experiments that I'm not going to show you here. It's actually a delayed binding of the ligand because of the differential conformation of the receptor due to this mutation. And we then asked the question, what we found in mice, whether it applies to primary human cells with this mutation. The answer is yes. So if we look here on the left, this is a simple experiment. If you take wild-type monocytes and you serum starve them, there will be about 20–25% of them that die, and no surprise there. But if you add the ligand for this receptor, you can actually rescue these by about 30 to 40%. Now, if you have monocytes that have that rescue through the system doesn't work. And we then asked the question, how does this work in primary human cells? Is this AKT dependent? The answer is yes. So if you take wild-type monocytes and you serum starve them, again, they have about 15 to 20% death. You can rescue that in the second bar with adding fractalkine. And the third bar is adding fractalkine in the presence of an AKT inhibitor, and you can see you can completely negate the effect. And the last bar is just the inhibitor itself, who has no effect. So it appears that in human monocytes, AKT is critical for mediating anti-apoptotic effects through this axis. And these primary monocytes that lack this, have this mutation, actually are unable to induce AKT after stimulation. So this axis appears to be completely impaired as a result of this mutation. It appears to have a profound effect on survival in ex vivo assays, but also clinically relevant in vivo, because if we take monocytes from these patients' counts of monocytes, you can see that these patients are actually monocytopenic, which adds this receptor alongside a few molecular factors as potential genetic risk factors at the population genetic level for monocytopenia. And with Melissa and John now at Duke, we're asking the next obvious question, whether, not at steady state as I'm showing you here, but whether those patients who were candidemic in the ICU and had this mutation, whether they were also unable to promote monocytosis during infection. So we don't have that data yet, but they're underway. Now, the second example I'm going to briefly mention about is about neutrophils, not monocyte macrophages. And we all know that neutrophils for Candida and Aspergillus, obviously, are the most potent antifungal agents. Patients with neutropenia are at risk for these infections. And when we looked at the transcriptional level in mice that are infected with Candida at different organs, this is a heat map where green means upregulation and red means downregulation and black means unchanged. In 4 different organs, as you can see at day 1, 4, and 7 after infection, all of these marked by a red arrow are neutrophil-targeted chemokine receptors, and they're significantly induced. And although we've done work on a few of those, today I'm going to talk about CXCR1, which is actually the first chemokine receptor ever cloned back in 1991. But for all of these 25 years, its functions were unclear because there were no knockout mice for this receptor. So generating mice and testing them in this model, this is the first phenotype attributable to CXCR1 deficiency. These mice, when they lack CXCR1, are unable to control the fungal burden on the right. They develop renal failure and they die. And our initial hypothesis, this being a chemokine receptor, was that this is a receptor that makes neutrophils come to the target organ. And we were wrong. As you can see here, there is no difference in recruitment of neutrophils in the infected tissue after infection. We went on to directly show that with mixed chimera experiments that I'm not showing you here for the interest of time. Instead, what this receptor appeared to mediate is the ability of neutrophils to damage Candida hyphae within the infected tissue. So on the left panel, you can see a wild-type neutrophil abscess in the kidney, where this is PA stain, and you can see a few elements of Candida still present form, but most of those elements have been destroyed. And on the right side, you can see, again, a kidney neutrophil abscess from a mouse that lacks CXCR1. And despite the presence of neutrophils, hyphae is marching through them as if neutrophils are not doing their job properly. This suggested to us that this is a receptor critical for killing of or damaging Candida by neutrophils. And indeed, that was the case when we sorted out neutrophils out of the infected kidney. They were significantly impaired in their capacity to damage Candida hyphae. That was not because the neutrophils had a problem with binding to the hyphae or internalizing pieces of hyphae, or their maturation or survival or oxidative burst or net formation were completely normal. Instead, what a major mechanism for destroying large extracellular particles besides the netosis is degranulation. And indeed, that was the problem with these neutrophils. So when you look on the left, there's a function of release of beta-glucuronidase, a major granule protein in neutrophils, as well as content of MPO. These were significantly decreased in the infected tissue of mice, in mice that lack CXCR1. This was actually a cell intrinsic defect, so it wasn't a function of the infected tissue, because when you take uninfected bone marrow neutrophils, they have exactly the same problem. So this is a cell intrinsic defect, and it also suggests that the problem in the degranulation is at least in part also related to granule formation, because these are not neutrophils that have gone out of the bone marrow yet. So we are working on trying to figure out exactly what these pathways are at the moment. Again, in the same kind of line of thinking, we looked for an allele in this receptor, which is 276. This is an allele that, as with the previous allele, is actually quite common in the general population. The previous allele has an allelic frequency of about 20%. This about 7-8%. And there were few studies showing that patients, children and adults who had this allele were otherwise healthy, but they had the propensity for bacterial renal infections, pyelonephritis. So we asked the same question in collaboration with our colleagues in Duke and Nijmegen, and we again found that in multivariate analysis, this was a risk factor for developing worse outcome defined as disseminated disease with an odds ratio of close to 4.5. Here again, we were able to test primary neutrophils from patients from healthy individuals with or without the allele, again through screening over 100 of those healthy donors from our blood bank. In this specific mutation, the problem, as opposed to the previous one, is that the receptor appears to be significantly decreased in expression. So as opposed to the binding of the ligand in the first example here, is that the receptor is not on the surface. And consistent with what we find in mice, you can see here that the human neutrophils from healthy otherwise people who have this mutation, and about 7 or 8% of all of us around us would be such individuals, are not able to kill Candida very well. And they have the same degranulation defect that the knockout mouse neutrophils had. Again, closing the loop, starting from a mouse, going to population genetics, and coming back to primary human cells showing the same mechanism. So, in summary, what I've shown you is that in this model, Candida gets into the infected tissue, in this case kidney, very, very quickly, within a couple of hours. Now we have intravital data to show that as early as 30 minutes, Candida is within macrophages in the infected kidney, which is quite striking to me. Now, this early interaction of macrophages and Candida is highly dependent on CX3CR1, which provides survival signals for these cells in order to engulf and kill Candida. Now, we have live imaging data over a few hours that clearly show that macrophages are very competent killing Candida in the infected organs. Not as effective as neutrophils, but certainly they do a pretty good job. Now, neutrophils come into the tissue early on, and we don't yet know by which mechanisms they do come. But when they are there, they use CXCR1 in order to damage Candida hyphae. And these two mechanisms for both of these receptors that I showed, that I mentioned in mice, do seem to apply to human cells as well. At the same time, monocytes come into the tissue. This is through CCR2. Tobias had a nice paper on this recently. And what he showed is that these cells only act within the first day or two after infection. And the reason really for that is what I showed you in the intravital imaging. These cells do not get into the tubules, and there's only a very, very brief window of opportunity for them to come into the tissue before Candida evades them by getting into the collecting system. So that's the reason why the CX3CR1 receptor system appears to be more important for. These are macrophages that are already in the tissue as opposed to CCR2 recruited monocytes that come into the tissue and have a much shorter window of time to interact with Candida. Later on, when neutrophils come further with more Candida in the tissue, they actually get into the tubules and they cause more disease and more tissue injury. And this pathway seems to be mediated mostly by CCR1. So we now have data with the Novartis CCR1 antagonist that you can actually cure mice from Candidemia if you give them this receptor at a time when neutrophils are coming late. So what I've shown you today first is that it's very important to interrogate immune responses at the site where it matters. If we had not looked at kidney macrophages, we would think that macrophages are really lousy in doing mediating effective host defense against Candida, and that's not the case. If we have not looked by intravital microscopy, we will never know that the only time they interact with Candida is the very first couple of hours. So if you're looking at lung models or brain models, it's important to try to address and not peritoneal macrophages, despite the limitations, obviously, and the technical problems of doing that. I think importantly, chemokine receptors and their ligand do way more than just telling a cell to go from one place to another. And I urge you to think of those molecules when you look at immunity in your models. And I think importantly, these are just two examples of how we can use bench-to-bedside approaches to perhaps develop individualized risk stratification strategies and prognostication strategies so that maybe in a few years from now, we'll be able to say that from these 100 patients who are in the ICU, those 5 or 10 are at much higher risk to develop it, so we should probably put them on prophylaxis or again vaccinate them or give them a more they took about 200 patients with Candidemia and 11,000 healthy people, and ask the question, those that were candidemic, did they have enriched mutations, SNPs that would put them at higher risk? And these are the three gene SNP mutations that they found, genes that were actually not known to mediate any role in immunity. They went back and provided some proof of concept that at the biological level, there is relevance to this. So following the reverse approach of what I showed you earlier, these again are also candidates for sitting down and really thinking of ways to design trials that would ask the question, can we develop prophylaxis strategies in the ICU based on immunogenetics? And I think the data are there and the time has come. So with that, I would like to thank Leka and Mandy, who did most of the work that I showed you today. Melissa and John from Duke, as well as Michail from Nijmegen, and you for your attention from a distance. And I think, unfortunately, I'm not going to be able to answer any questions, but you can all email me if you had any burning questions from what I said now, or tell Pete to communicate those to me. Thank you so much.
Thank you, Michail.